My name is Caitlin, and I am a research associate for the Core Surfaces of Glyconet located in the Department of Chemistry at the University of Alberta. Today we will be talking about the 2D NMR analysis of carbohydrates. Two-dimensional NMR can be useful when there is a mixture of compounds in your sample, or if there is complex coupling in your one-dimensional NMR spectrum, or if your sample is an oligosaccharide. There are a number of different types of two-dimensional NMR experiments, and today we will discuss the proton-proton COSY, the proton-proton TOCSY, the proton-carbon HMQC, and the proton-carbon HMBC. We will start with an overview of the type of information you can obtain from each of the different types of two-dimensional NMR experiments, and we will explain how to read each type of spectrum using the examples of known compounds. At the end of this module, I will take you through an example of how to characterize an unknown carbohydrate using all of the previously discussed types of NMR experiments. A COSY spectrum, or correlated spectroscopy, is a two-dimensional NMR experiment that measures the spin-spin coupling between neighboring hydrogens. This experiment is useful for when there is overlapping signals or when there is extensive second-order coupling and the coupling constants cannot easily be determined. Practically speaking, when we look at a COSY spectrum, we see that we have the 1D NMR spectra running along the x and y axes. There are two types of peaks in the COSY spectrum, peaks that run along the diagonal and cross peaks. The peaks that run along the diagonal represent the correlation between protons that resonate at the same frequency, i.e. peaks with the same chemical shift. The cross peaks are representative of correlations between two protons that each resonate at a different frequency or different chemical shift, but are coupled to one another. When interpreting the COSY spectrum, we are most interested in the cross peaks, as these peaks will typically give us information about which protons are interacting with one another up to 3 chemical bonds away from one another. To read a COSY spectrum, find a peak along the x-axis and draw a dashed line vertically. All of the cross peaks along that vertical line are protons that are coupled to your peak of interest. Looking along the vertical dashed line that we have drawn, we see that there is one cross peak present. If we follow that cross peak with a horizontal line until it intersects the diagonal, we can find the peak that is coupled to our proton of interest. Looking at the spectrum of this protected monosaccharide, we would typically expect the most downfield peak between 4.5 and 5.6 parts per million to be our anomeric proton. However, if we look at the most downfield peak in this spectrum at approximately 4.9 parts per million, we see that it is coupled to one other proton, which is also split into a doublet. We know that from the one-dimensional NMR that the hydrogen in the anomeric position should be a doublet, but the proton in position 2 should be a doublet of doublets due to the two chemically inequivalent hydrogens that neighbor it. For this reason, we know that neither the peaks at 4.9 or 4.6 parts per million are due to our anomeric proton. Moving to the next peak in the spectrum, we see a doublet at approximately 4.2 parts per million, which, if we follow the horizontal line to where it intersects the diagonal, we see that it is coupled to a proton that is a doublet of doublets at approximately 3.49 parts per million. Looking along the vertical of the peak at 3.49 parts per million, we see that there are two cross peaks, one at 4.2 parts per million and one at 3.59 parts per million. We know that the cross peak at 4.2 parts per million is due to the correlation between the blue and red proton. So our next peak of interest, the proton at position 3, must be the peak at 3.59 parts per million, as indicated by the green color. Looking at the vertical of the peak due to the green proton, a cross peak at 4.0 parts per million is observed and determined to be the hydrogen at position 4, as indicated by the purple color. The vertical of the peak at 4 parts per million indicates coupling interactions with a peak at 3.61 parts per million, which is the proton in position 5, as indicated by the orange color. The vertical of the peak due to the hydrogen at position 5 has two cross peaks, one at 4.0 parts per million, which we previously determined to be due to the position 4 proton, and one at 3.77 parts per million, which is due to the two protons at position 6. With the peaks of the carbohydrate skeleton identified, we see that we have 4 peaks left over. Looking at the structure of our compound shown on the right, we can deduce that the two doublets at 4.9 and 4.6 parts per million that each integrate to 2 must be due to the benzyl protons, and that the singlet observed at 3.55 parts per million with an integration of 3 must be due to the methoxy group. A TOCSY, or total correlation spectroscopy experiment, has many similarities to the COSY. It is a measure of the correlation between protons through spin-spin couplings, or through bond couplings. The big difference between the TOCSY and the COSY is that the TOCSY will show correlations throughout an entire spin system, whereas the COSY is limited to correlations that are up to 3 chemical bonds or less away. A spin system is defined as a set of nuclei that share no couplings with its outside environment. This means that regardless of how far away a hydrogen is, it will couple to all other protons within the spin system. A spin system is interrupted by heteroatoms and quaternary carbons. The TOCSY experiment is extremely sensitive to something called the mixing time. This is the amount of time the experiment allows for magnetization transfer, allowing you to see the through bond couplings. If you are not seeing all the proton signals within a particular spin system, your mixing time may be too short, and you should talk to your NMR specialist about extending this particular parameter in the experiment. Because the TOCSY experiment has the ability to show all correlations within spin systems, the TOCSY experiment is particularly useful for oligosaccharides, as the backbone hydrogens of each monosaccharide are in an isolated spin system. To interpret the TOCSY, we can again draw a line through the diagonal and see that each peak couples to itself. In addition, if we look along the vertical of any peak, we see many signals that indicate coupling throughout the entire spin system. Because it shows coupling throughout the entire spin system, it is hard to definitively assign chemical shifts to specific hydrogens within the molecule. A COSY becomes much more useful to walk along the backbone of the sugar. A TOCSY becomes useful when you have an oligosaccharide and you can isolate peaks within each monosaccharide within your oligosaccharide and then turn to the COSY to definitively assign the chemical shifts to specific hydrogens. The HMQC, or the Heteronuclear Multiple Quantum Coherence experiment, is a two-dimensional NMR experiment that will give direct one-bond couplings between protons and carbons. In looking at the spectrum, you can see that there is a proton spectrum along the x-axis and a carbon spectrum along the y-axis. There is no diagonal in the HMQC spectrum. Rather, each crosspeak in the two-dimensional spectrum will give you information on the direct carbon-hydrogen linkages within your molecule. Because quaternary carbons have no hydrogens attached, they do not appear in the HMQC spectrum. To read an HMQC spectrum, find a carbon peak of interest and draw a dashed horizontal line. Where you find a crosspeak in the spectrum, draw a vertical line. The carbon and proton peaks that are now connected by the dashed lines are directly connected within your molecule. In this example, I first found the anomeric carbon, identified by its unique chemical shift in the carbon NMR, and located its crosspeak along the horizontal. By following it upwards with a vertical line, I have now identified the proton at position 1. Note that there are other interesting features that you can see within the HMQC. If you look along the horizontal of the carbon peak at 60 parts per million, you see that there appears to be 2 cross peaks. This indicates that there are 2 hydrogens that are attached to the same carbon, most likely the CH2 at position 6 in most sugar molecules. We also know that carbons which are adjacent to nitrogens have lower chemical shifts at about 50 parts per million. We can identify the proton at position 2 by following the horizontal and vertical of the carbon that we have identified as being next to the nitrogen. You may have also noticed that there is a proton peak that has no cross peak in the 2D HMQC. This is due to the fact that the proton is attached to the nitrogen of the N-acetyl group rather than a carbon. In fact, if you ran a proton nitrogen 15 HMQC, you would only see one peak, which would be the correlation between this proton and its corresponding nitrogen. The HMBC, or the Heteronuclear Multiple Bond Correlation experiment, gives correlations between carbons and protons that are separated by 2, 3, and sometimes even 4 bonds apart. It differs from the HMQC in that direct one-bond correlations are suppressed. The connectivity information that is obtained from this experiment is analogous to the information that is obtained from a proton-proton COSY. It is important to note that the absence of crosspeaks does not confirm a carbon and proton pair is far apart. This is because in proton-carbon pairs that are 3 bonds apart, the intensity of the crosspeak follows the Karplus equation, which was discussed in the 1D NMR module. HMBC is particularly useful when deducing the location of protecting groups or substituents that are present on the hydroxy groups of a sugar. It is also useful in determining the linkages between different monosaccharide units. If we look at the example on the right, we can draw a vertical line at the proton signal, and the cross peaks present will indicate the carbon atoms that are up to 3 chemical bonds away. Looking at the vertical of the anomeric proton, we see that there are 3 cross peaks, 2 of which are correlations to the backbone carbons, and 1 of which is a correlation to a carbonyl group. This indicates that the proton at position 1 has an acetyl protecting group. In the same way, we can look at the vertical of the proton at position 2. We see that it also couples to a carbonyl, and we know from the HMQC that the carbon at position 2 is adjacent to a nitrogen atom. So by using the information from the HMQC and HMBC, we can deduce that this sugar is N-acetylated at position 2. A similar analysis can be done for the proton at position 3 and the proton at position 4, each indicating that the hydroxy groups are acetylated. Let's look at an example of how we can use all of these NMR experiments that we have discussed to determine the structure of an unknown sample. If we look at the 1D proton, we see that there is one region of the spectrum that is well defined and has clear coupling constants, and a second region of the NMR that has extensive overlap of peaks and unclear coupling constants. In the 1D carbon NMR, we see that many of the carbon signals are clumped together and only the signals due to the anomeric protons are clear. It is important to note that the presence of 2 signals in the anomeric region of the carbon NMR, because this tells us that we either have a mixture of alpha-beta anomers or that we have a disaccharide. To identify the proton peaks that are due to the anomeric position, we look at our HMQC. We draw horizontal and vertical lines that connect each carbon peak with a proton peak in order to determine the chemical shift of each proton in the anomeric position. With the anomeric protons identified, we can move to the TOCSY experiment to determine which peaks are due to which monosaccharide present in the sample. If we draw vertical lines to the proton in the anomeric position and then follow the horizontal for each cross peak observed, we can identify and isolate the peaks in the 1D NMR that are due to each individual monosaccharide. Once this is done, for each monosaccharide present, we have an outline of the sugar backbone for each component. Having identified which peaks belong to which monosaccharide, we can turn to the COSY to definitively assign chemical shifts to each proton present. If we start with the monosaccharide that I have labeled as A, we can walk along the backbone, identifying protons one at a time. Ideally, in a COSY spectrum, each proton peak should have a discernible cross peak for each neighboring hydrogen. However, sometimes the cross peaks are unclear or hard to distinguish. We will begin by drawing our diagonal line, which is shown here in black. Next, we draw a horizontal line at the first anomeric proton, which I have labeled as HA1. We see that there is a cross peak at 3.3 parts per million, and by following the vertical upwards, we are able to identify the proton at position 2, as indicated by the purple color. If we look at the other cross peak that is present in the vertical of H2A, we can draw a horizontal line that intersects the diagonal at 3.63 parts per million. By drawing a vertical line upwards at the point of intersection, we can identify H3A as indicated by the blue color. You will notice that along the vertical of H3A, there is only one cross peak, which is due to the proton at position 2. However, by looking along the horizontal of H3A, we can see that there is a cross peak at 3.82 that corresponds to the proton at H3A, indicating that this peak is due to the proton in position 4, as indicated by the orange color. Proton 5 is identified by following the second cross peak along H4A's vertical to where it intersects the diagonal at approximately 4.66 parts per million. Looking at the vertical of H5A, a second cross peak is observed, and by following the horizontal to where it intersects the diagonal, proton H6A1 can be identified at 3.62 parts per million. In the same manner that H6A1 was identified, H62A can be identified at 3.66 parts per million. Having identified all the peaks that are due to monosaccharide A, we can repeat the process for the peaks in monosaccharide B. Once again, we have our diagonal shown in black. A horizontal line is drawn at the peak H1B until a cross peak is observed. Drawing a vertical line at the cross peak, we can identify proton at position 2 as having a chemical shift of 3.33 parts per million. Looking along the vertical of H2b, we see an additional cross-peak which intersects the diagonal at 3.51 parts per million. By drawing a vertical line at the point of intersection, proton 3b is identified and shown in blue. Again, the vertical of H3b only has one cross-peak. However, if we look along the horizontal of H3b, we find a corresponding cross-peak at 3.75 parts per million, indicating that this is the proton at position 4, as indicated by the orange color. Proton 5 is identified by following the horizontal of H4b until it intersects the diagonal at 3.84 parts per million. Drawing a vertical line at the point of intersection identifies proton H5b as indicated by the purple color. Following the second visible cross-peak present along the vertical of H5b until it intersects the diagonal distinguishes proton H6b1 as shown by the light blue color. H6b2 is identified in the same way and also shown in the light blue color. Now that we know the specific chemical shifts for each proton in each monosaccharide unit, we can use the coupling constants to determine the relative stereochemistry of the hydroxyl groups and to determine the identity of each monosaccharide in the sample. This topic was covered in our module on 1D NMR analysis, so if you would like step-by-step instructions on how to use coupling constants to determine your monosaccharide identity, please refer back to that module. Let's consider the monosaccharide that I have labeled as A first. Based on the large coupling constant between H1A and H2A, we know that they must be in an axial-axial relationship to one another. The additional large coupling constant between H2A and H3A also indicates an axial-axial relationship. Again, a large coupling constant between H3A and H4A indicates an axial-axial relationship between those two protons. H4A has an additional second large coupling constant, which indicates H5A is also in an axial-axial relationship to H4A. The coupling constants are difficult to distinguish between H5A and H6A1 and A2. However, it does not impact our ability to determine the monosaccharide identity. Based on the coupling constants for monosaccharide A, we can determine that this unit is a glucose unit. The same process can be done for monosaccharide B, and in this case, it is also determined that monosaccharide B is a glucose unit. So far, we have used the HMQC to identify our anomeric protons, and the TOCSY to identify which peaks belong to which monosaccharide unit. The COSY was used to assign specific chemical shifts to each proton, and then the coupling constants were used to determine the identity of our monosaccharide units. The last step is to determine whether or not the monosaccharides are linked together to form a disaccharide, or if the sample is a mix of alpha-beta anomers. The HMBC is particularly useful to determine linkages because if a cross-peak is present between two monosaccharide units, we know that they must be joined by a chemical bond. If there are no cross-peaks present between the monosaccharide units, then we know that the monosaccharides are not linked together and the sample is a mixture of individual monosaccharides. If we draw a horizontal line at each of the anomeric carbons labeled C1A and C1B, and then follow any cross-peaks with a vertical, we can determine if any correlations exist between the two monosaccharides. In this case, we see a correlation between C1b and H3A, C1b and H3b, and C1b and H2b. The important cross-peak here is the one between C1b and H3A. This tells us our two monosaccharides are linked in a 1,3 linkage. Using all of the information that we have gathered, we are able to determine that our unknown compound is beta-D-glucopyranose 1,3-beta-D-glucopyranose. Today we've talked about 4 different kinds of two-dimensional NMR experiments that can be used to characterize complex carbohydrate samples. The COSY, which gives proton-proton through-bond correlations up to 3 chemical bonds away, the TOCSY, which gives proton-proton through-bond correlations for an entire spin system, the HMQC, which gives direct carbon-hydrogen 1-bond correlations, and the HMBC, which gives us proton-carbon through-bond correlations that are 2 to 3 bonds away.